So it seems like there has been some problems with the virtual lab for the flower dissection. I'm able to pull it up on my screen. So what we'll do is I'll just upload this recording for you to be able to go through the lab, and you'll be able to answer your questions and also just turn in that you have completed it. Because I'm not sure what exactly happened. Sometimes it takes a little bit just to load, but a lot of the issues stem from this little puzzle piece right up here. With that puzzle piece is your flash player, your extensions, and your flash. So even if you can't get that, we'll just run through this. So you should read the introduction. So plants are marvelous magicians. They can use the same set of basic materials to build a variety of floral structures to fit their own specialized needs. Ready to see the magic? Let's join the interactive virtual plant dissection lab now. So in this course, you're going to learn about the basic terminologies of flowers with well-illustrated information, and then actively participate in the virtual experiments by deciding the dissection methods, judging the type of structural features exhibited by the specimens, accompanied with interactive visual references. Now, I do want to say that this is brought to you by these people here, and they are the ones that are responsible for making this virtual lab. For your dissection manual, it's important for you to know the different types of plant parts and what their functions are. Flowers have many features that are extremely useful in plant identification and taxonomy, so we can look at the color, the number of petals, what the reproductive organs, like how many were they located. So we're able to use those in order to identify a plant. So before we do that, we need to first learn about the major reproductive organs of a flower. The first is the sepal, which is the outermost layer that encloses the flower bud before it opens. They're little green leaf-looking structures that protect the flower before it opens. You then have your petals that are showy and brightly colored in order to attract pollinators, and it's just really their whole job is to sit there and look pretty. You have the tepals, referring to the petals and the sepals, which look similar and are indistinguishable in appearance. We then have the stamens. It's the male part of the flower. Consists of anthers and filaments. The filament is the slender stalk that supports the anther. The anther is where the pollen grains are produced. So we look at this picture here. You can see the stamen. Here we have the filament, which is the stick-like structure, and then we have the anthers here, and the pollen would sit on those anthers. The pistil is the female part of the flower, which is in this picture here. It is a vase-like shape that bears the eggs that called ovules. And the ovary is connected to the stigma by a thin stalk called the style. Stigmatic surface functions to catch the pollen grain. So remember that every flower is unique. You can invent your own way to investigate the flower of interest. You don't have to just go in those specific ways. All right, so we look at this lily here. We're looking at what's called the morphology, or the way the plant is set up from top, side, and bottom views. So we're going to try and identify the basic floral organs, record the numbers of floral organs whenever available. So we have the tepal, the stamen, and the pistil. From our top view, we have your tepal, which is the petals and then those early leaves that protect the bud. You have your stamens, and then your pistil. If we were to look at that from the side view, we have the tepal, the stamen, and the pistil. Bottom view, we can see this tepal here. It's right here. You can see those early leaves that protected that flower while it's blooming. It's indistinguishable from the petal. So if we look at the arrangement of the floral parts, the tepals are on the outer whorl. If we took apart this lily, they'd be the first three. And then we have our, in the inner whorl, our next removed. And then we just have the male and female parts. This receptacle of the female part is down here of the pistil. Your stamens are here. And then there's your petals. So as you dissect a flower, what you should do is cut along a transverse plane at a point just above the receptacle. So you cut down through, and then you're able to see the different parts of that flower. So here we have the female part, the male, and then your petals. So here's your female part of the pistil, here's your male part, and then again, the petals or tepals in this case, inner whorl and outer whorl. Whenever you dissect, you want to isolate the stamens from the receptacle. You remove those, and in this case, there's 6. Then your receptacle is along the bottom. So what we're looking at is just the male part. So this is all the stamen, immature and mature. You have the anther, and then what's known as a filament. For your pistil, you have, this is the female part of the flower. You have the ovary, style, and stigma at the top. Stigma is the sticky part that catches the pollen. The pollen will travel down the style, it's a tube-like structure, down into the ovary to fertilize the eggs. We look closer at the stigma. It is sticky, and on the lily you can see 3 lobes that can be observed, and that's what catches your pollen. In order to do a cross section, you want to isolate the ovary, gently cut along the transverse plane to generate a cross section. Notice they're being very careful, and it's very thin. So we pulled that up on the microscope. We can see here's your seeds, the oval, the placenta, and then your ovary wall. Now if we did a longitudinal section, which is a long cut, you would carefully cut along the edge, getting your samples, and then we would be able to see this from alongside. Here's your wall, your oval or immature seeds at this point, and then the placenta that's holding those seeds in place. For your basic terms, if we look at another diagram of the same thing without looking at a real flower, we can number them 1 through 4. 1 is the carpel, 2 is the stamen, 3 is the petal, and 4 is the sepal. From a flower standpoint, you have the receptacle down here, which is where it holds the flower. Then you have the ovary, which holds your seeds, or the oval. The style is the neck of the vase, and then we have the stigma. We then have the male parts, the pollen, anther, filament, corolla, calyx, and pedicle. And they do have some fancy names for this. The female part of the flower is the Genesium, and then we have the Androcium for the male part. Then these down here, the petal and the sepal together, is the paraneth. You don't need to exactly know those terms, just be familiar with them. Like I said, I use this dissection lab also for my high school floriculture class, and so there is some higher level things in here that I just want you to know exist, but you don't need to absolutely know and memorize them. Like this tab, you should just know the basic parts of the flower, not the fancy names and things like that. Now your ovaries can have different positions based on the flower. So we have, this is called Ovary Superior. That means the ovary is larger. The flower is hypogenous. For example, we look here, ovary takes up the most space. Right? Ovary Superior means that it takes up more space. Everything else is smaller. We look at Ovary Superior, flower Epigenesis. We can see that everything is pretty well equal here. You have Ovary Inferior, flower Epigenesis. So the ovary is enclosed and you can't really see it. We have ovary half inferior where it's only half covered. Placentation, you have these different things. They have different looks to them. Then we also look at the stamen arrangement as well, the male part arrangement. Is it enclosed down inside the flower? Are they free range, free flowing? Are they encased down below? Are they spread out evenly? Are they up along the entire pistil? Are they long and flowy like this? I mean, it just depends on your type of flower. Now, if we actually went in now, we can go in and look at some different flowers. The way you would navigate this is you click to enlarge, and you can look at the different flowers, right? So if we look up here first at the male part of the flowers, we are wanting to know how many stamens we have. So you would click on the knife, and it would cut it open for you, and you're able to zoom in and look at these. So if we look at the male part of the flower, we have the stigma, you have the style. You can see that it has a little bit of texture on here. That is what holds the pollen. Look at the flower here, this petal. It has texture, it's not just smooth. And we look down here, we have the male part of the flower. I'm sorry, I misspoke on here. All right, I misspoke. This is the top of the female part of the flower. Misspoke. Because down here we have the male parts down here. So you may be thinking, how in the world does this flower get pollinated? This flower would need to be pollinated from another flower, not itself. Some flowers can be self-pollinating, while others require the pollen from another plant. So right down in here we have some hair-like structures that would help hold in pollen. And we do have some pollen spread out down through here, but here we have our male part, so we have our filament, and then our anther, and the pollen would sit here. This is the style, or the neck of the vase, that would carry up and over. If we go to the second flower, we're looking at we have one carpel. Let's look at the differences. We're going to want to look at different views. So we're going to look at the Genesium. Here it is close up. We look at a longitudinal section. This is where we cut it long ways. So you can see you have your immature eggs, you have your placenta, and then your ovary walls. A transverse section, that's where you take it and cut it into different layers. And you can see your different parts here. Here's your male parts of the flower. As you can see, this is the female part of the flower. This is the male. Different flowers have different structures. So from up above, you can see the female part, the male parts. This is a magnolia. You go on down, we have another type of flower. We click to enlarge to explore the flower. We have 3 different areas that we're going to be looking at. We're going to look at the flower. So this is the whole flower, side views at different angles. And here we have our different parts. Your sepals are down here, your petals, the pistil hangs here. Then you have your male parts. If we were to conduct this cut, we can see our different levels of dissection in the different parts. If we looked at the female part, we can see, let's look at the transverse section first. You can see here's your ovals, here's your placenta, and here is the ovary walls. If we were to do a longitudinal section cut, you can see the same structures. Personally, I preferred the longitudinal cut. I think you're able to see the structure more wholly, but then again, also transverse section also has its benefits as well. This is a fertilized ovary after flower wilting. If we were to cut that, now we can see the differences in the seeds. Here we have the sepal, we have the bract, and here are our ovules that have now been fertilized. Cut it again, and we can see the seeds are fuller, they're starting to get more broad. You can see the difference between them. These still aren't fully mature seeds. They would be a different color, but they are fuller. They're developing into a there we go. I had to answer a phone call there, so it disconnected me. So we can look through here. We can look at the different things. We have a leaf with a broad blade. You have a developing fruit, and we've explored the flower. So this was just designed to be a quick virtual lab for you to see some different examples of flowers and how their parts may look a little different, but they all contain the same sort of parts. Now, some plants have specifically male flowers and female flowers, in which case then that flower would only have those parts specifically. It would either have the pistil or the anther, or stamen rather. It wouldn't have both. In the flowers that I had for us to do in class, they would have had both of those. We'll see if we get into class next week. Have the plants they're holding on. I mean, they're starting to look a little rough, but if they're still usable and we're in school next week, I'll bring them in for you to at least look at and notice the parts for them. But if you have any questions, please don't hesitate to reach out to me. But this should help you with that actual virtual plant lab. So if you can't get it to load, that's what we're going to do with it.